So thank you again for taking part and coming to listen to me. I'm going to try and present some of my research, and that is focusing on enzymes that can help degrade and reutilize what we call recalcitrant waste biomass. And this picture also shows that I do a lot of crystallography, for those who would in the know. I think in tradition with previous talks, I'll try and give a little bit of background about myself. For those of you that do not know me, I'm Italian. I was born in the same year as the Protein Data Bank, so that has had an anniversary this year, so you know how old I am now as well. I am Italian, but I have been educated in the UK. I did a degree in biochemistry, several exchanges, one in Cambridge and one in Germany, and my PhD in Reading, but associated with the University of London. And then I came here and I sort of stuck. I must have liked both Copenhagen and CHEM, because I've been there since my postdoc with Professor Signe Larsen. And I'm at the moment head of the biological chemistry section, although to be fair, people in the biological chemistry section head themselves most of the time, but formally at least. And also I would like to add that a special connection, at least in my head, with Japan. I've been there in exchange many times, and I am currently I've been a fellow for the Japanese Society for the Promotion of Science, and I am now chairperson of the Alumni Club Denmark. So if you have been a JSPS fellow or are interested in becoming one, I can plug the Alumni Association at the moment. And in previous talks, people have also said a little bit about their thoughts about life in academia. So I'll give you just one piece of my own impression. Life as an academic scientist is quite frankly at times a little bit hard, but when it's a good day, then you remember that you are very privileged in working with a lot of people from all over the world, for the most extremely intelligent people, and sometimes also very nice people. So what I'll focus on mostly today are also collaborations that I've enjoyed immensely also as a person. Just to give an idea of what we do in the biological chemistry section in general, we work, as the blurb on the homepage says, on the structure and function of the molecules of life. And by this we mean we do a lot of work with the proteins, with DNAs interacting with the drugs, for example, proteins interacting with DNA, proteins interacting with the metals and with polysaccharides. So if you want to know more about also about the section and who else is in there, you can have a look at the homepage as well. But today I'm going to focus on something that is very much focused on at the moment. Now it's actually fashionable, but I've worked on these sort of problems all my scientific life. And that's the idea that we can turn waste products, especially waste product that is rich in biomass, in what I call recalcitrant biomass, particularly rich in cellulose and hemicellulose, so things like what's left after making beer, potato peels, spent coffee grounds, whatever that contains this type of biomass. And in principle also that applies to other recalcitrant waste, and somehow have find ways to convert it into useful stuff. And that, of course, is not just a solution that will come from science and technology. It needs to change our consumer habits. There's politics, there's money involved in it. But the idea is basically how to convert waste containing valuable chemicals into energy of various forms, bulk chemicals that can be used for synthesis, fine chemicals that maybe have a value in themselves. Of course, we must always remember that as well as the high-tech solutions for these conversions, there's also the low-tech solution. So there's a concept of upcycling, there's a reuse of waste. That's me upcycling some spent coffee grounds from my coffee filters, makes an excellent face mask, makes also a mess in the bathroom. So in case you want to try, I've warned you. But I've talked about recalcitrant biomass, and if we look in the Oxford Dictionary, recalcitrant, this is a word that's used very much in this field, but maybe it's not clear to you what I mean. But the definition is having an obstinately uncooperative attitude towards authority or discipline, which seems a weird thing to attribute to biomass. Normally, we would attribute it to things like donkeys or mules. My mother may come with this example, perhaps, that fits the bill. So what do we mean when we're talking about recalcitrant biomass? We are talking about biomass that's derived primarily from the plant cell wall. And the plant cell wall actually is treated quite unfairly when we call it recalcitrant, because the function of the plant cell wall is obviously to protect the plant, so it does its best not to be broken down easily and attacked and made into smaller bits. So when it's being recalcitrant, it's actually just doing its job. But when we want to recycle stuff into plant cell wall containing biomass, which is an awful lot of it, into useful products, so then of course it's in our way. That's why we call it recalcitrant. So this is a sketch of a primary cell wall, and it gives you some idea perhaps of why it is difficult to attack it, both with chemicals and with enzymes. We have microfibrils of cellulose here, which are in themselves quite hard to digest because of their crystallinity, and there are complex networks of other polysaccharides that make this all into a bit of a mess to get into. If we are talking about plant cell wall which also contain lignin, it's even worse because, as you can see in this nice figure that I've taken from Nishimura et al., you see the cellulose fibrils here in the wood, and they are surrounded by this hemicellulose, which is wrapping around them, and attached to that covalently, we even have lignin. In a sort of homemade sketch here, we have cellulose fibrils in blue, and they seem quite innocent if you think that cellulose is beta-1,4 linked glucose residues. So, however, since they're packed into a crystalline structure, they're quite difficult to access. What I'm shown in green that is connecting them is this hemicellulose network, which primarily I will be talking about the hemicellulose that is based on a xylan backbone. Again, quite innocent looking, beta-1,4 linked xylose. However, a lot of branching heterogeneity, which depends on the type of plant cell wall, on the time of the year, etc., etc. And covalently linked to this, we have the brown stuff that is the lignin, which is, if you want, it's an amazing source of all sorts of possible structures. This is, of course, just some sort of sketch. It's even more complicated, but it's, again, very difficult to get to and separate. So when we say recalcitrant biomass, we are talking about primarily biomass that has a lot of plant cell wall residues in it, and it's recalcitrant to degradation because that is the function of the plant cell wall, and it's hard to penetrate for chemicals and enzymes, chemically and structurally heterogeneous and interconnected because it's a set of networks of polymers that are interconnected. We have also the crystallinity of cellulose to deal with. So we have this conundrum. We have all this difficulty of getting into the plant cell wall based biomass, but it's a treasure trove of chemical substructures if you really could extract them. And that is why that is the conundrum that we need to solve if we really want to utilize it to the full. And I'm not dogmatic. I think that we should use anything that goes. So of course, enzymes are just part of the solution. And then I think that they are particularly part of the solutions that are in the second strategy that you could approach this problem with. And the first strategy is the most used, I think, at the moment, because it's the most useful logistically. I call it small, simple bits. That is, you try and chop and degrade the plant cell wall by chemical or enzymatic methods into, for example, glucose that you can ferment to biofuels, bioethanol, for example, and to make chemical feedstock. So anyway, try and get some small parts. But if you take the strategy that is called big complicated bits, in these fancy words that I have devised for this talk, then you are trying to utilize the substructures that are already in the plant cell walls and, for example, use them for ingredients in the cosmetic industry, in the food industry, for medicines, etc., etc. Yeah, so I will be showing a lot of protein structures, which will be determined by crystallography, and some of you are very familiar with crystallography to solve structures, but usually they are smaller. So just in case you're not familiar with this, we will have electron density, which is quite complex, and it can be quite hard to absorb, especially if you're not used to it. Even if you just show the non-hydrogen atoms, protein structures can be quite overwhelming. So these dots here now are water. So often we don't show all the waters. Maybe we show only the ones that we are interested in. And we often just show the backbone that we can see here as a cartoon, and then just show the interesting side chains on that. At the same time, I want to take the opportunity to thank all the facilities where we collect data, primarily synchrotron facility and DANSCAT that funds us travel there. So just quickly, we've been working for a long time on a class of enzymes that is called the Lytic polysaccharide MonoOxygenases, and these are copper enzymes with a fairly debated mechanism. We've been funded by the ERA-IB network a while back, and now by DFF and Novo Nordisk Fund to work on these enzymes, which I will only talk about for a couple of slides. But they basically are metalloenzymes that can activate either molecular oxygen or more probably hydrogen peroxide in most cases to break down the glycosidic linkage. And this is a figure that Tobias Tandrup made for this review. And this is a little video that Christian Franzen, who was a previous PhD student in the group, made. And we show how we contributed to this field by basically understanding how these enzymes bind to the cellulose chains structurally. And this is actually very important because it gives us a clue of why these enzymes are particularly good and useful in attacking crystalline cellulose. And this is an animation that was made by Tobias, where we can see that if we have crystalline cellulose, because these enzymes have this flat surface that you've seen that the cellulose chains can go onto, they can then place themselves here, and without having to distort the sugar chain, they can actually break it. That means that other enzymes that need to wrap around the sugar chain in order to be able to cleave the bonds, they have better access. So in this way, they've been quite interesting to study and only emerged in the last 10 years. So that's a contribution we made to that in a couple of slides, because today I really want to focus on some other enzymes that we've been working on. A fantastic collaboration with a group of Johan Larsbrink at the Chalmers University of Technology, which I've enjoyed really a lot. And his postdoc Scott, who's been excellent in this, and the 3 PhD students, Jenny, Dan, and Andrea, that have been involved. And then on our side, apart from me, Jens Kressin, Navarro Paulsen, when he was still doing research, has been involved, and postdoc from China that brought some new methods to the group, Ziyu Zhang, and then which I see is here twice, but then a lot of other students have been and are involved in this at the moment. And this has been funded by a small Interreg grant at the beginning, and the Novo Nordisk funding now more recently, with the grant recently expired. So we are desperately looking for new money to continue this. But anyway, this is the diagram we saw before, and these enzymes can attack some of the bonds that link the hemicellulose to lignin. So if we can separate the polysaccharide part from lignin better, the idea is that we can probably better utilize the substructures that are in the polysaccharide part and the lignin part for the plant cell wall. And these glucuronoyl esterases, if you are into enzymes, into carbohydrate active enzymes, they belong to the carbohydrate esterase family 15 in Casey. But in general, they belong to the much bigger class of alpha-beta hydrolases, which is a large superfamily with a conserved fold that contains a lot of interesting activities also from industrial point of view, proteases, lipases, etc. And they have a very classical conserved catalytic triad, the serine hydrolases, so they have a serine histidine acid type of catalytic triad. As I said, just to help you orient with the structures, I know that not all of you work with proteins. This is the basic structure as outlined in some key old papers of the structure, common structure of alpha-beta hydrolases. And we have this long beta sheet here with a slight anti-parallel part here, and then these 5 parallel beta strands here. Where we have the catalytic nucleophile, the serine, at the end of beta strand 5, the acid at the end of beta strand 7, and the histidine in this longer loop that goes towards the C terminus and is followed by a helix. And then we have all these 4 helix that are under these last beta strands here in the sheet. Just to see what it then looks like in the glucuronoyl esterases, we can see here one of the first structures that was determined, one of the fungal members of the family. And we can see here you have this long beta sheet. It's not quite as nice and neat as in the picture, of course. It's actually longer in glucuronoyl esterases, but we can see the catalytic serine here at the end of beta strand 5. And we can see the catalytic histidine, which is in this longer loop. You can see in both the diagram and the actual structural diagram. And then in the classical alpha-beta hydrolases, the acid is actually here, where this asparagine is in the glucuronoyl esterases. So in this glucuronoyl esterase, in the fungal ones, we have the acid actually placed here at the end of beta strand 6. And then just to help you orient in this, you can see the 4 helices that are down here. Now they are highlighted also in the structural diagram. And we have then also this long helix here at the C terminus that's coming up. Yeah. So when we started the project, and this is, I will not go into this, but this is a tree that relates that gives a representation of the similarity between the sequences, the primary structures, the sequence of amino acids in all these members of the C15, the carbohydrate esterase 15 family. And if you know how to read this by the length of the branching, you can see how close sequences are to each other. But roughly speaking, we have sequences from fungi and sequences from bacteria on this side and on this side, respectively. And when we started the project, it was basically mostly characterized fungal sequences, especially in terms of structures. And this is where we started the project. While we started the project, then this structure of a bacterial enzyme also was determined, and then came our contributions, our first contribution, which I think is a very important one, because we both characterize the functionally 10 different enzymes in the family in parallel, but also 2 of the structures. Then we have determined the structure and also functional characterization of this more distant bacterial member, and finally, most recently, of a hyperthermophilic bacterium. I mean, this thing is stable at like 80-90 degrees, so it's quite remarkable. And why do I think that is particularly important? This first paper we had, where we had this parallel functional characterization. It's because even though it's a characterization of on a very simple substrate, so they're supposed to be a model for the plant cell wall polysaccharides, but you obviously see this is very simplified. This is also a generic esterase substrate, so it's not even trying to mimic any substructure of the plant cell wall. What we can see is some clear differences in the members of this family in terms of their specificity for substructures of the plant cell wall. And you're not all familiar with this probably, but this is Kcat over Km, which is a common way of characterizing the kinetics or conversion by enzymes on different substrates. So this is what is normally called catalytic efficiency on a particular substrate. And let's try and get the laser pointer back up, perhaps. Just to try and make sense of this data and try and tell you why I think it's significant, is we have, for example, members of the family. So these are different enzymes, right? That have been produced and characterized by Johan in Johan's group. And you can see that these ones are pretty much active on all the substrates that have been thrown at them, with a little bit of differences, but generally speaking, they are quite active on all of them. This is just lousy on everything. These ones have quite good activities on the substrates that have galacturonic acid as their base, and the esters of glucuronic acid, but they are not active, almost not at all, on the ester of galacturonic acid that we have here. This one is active on these 2, but not on these. So not only it needs the sugar part to be a glucuronic, a glucose, it also needs a bulky group here. So we can see that in order to be able to use the correct enzyme for the correct type of biomass, we're going to need some fundamental knowledge here. And I can see that I'm going to run out of time. So I'm going to go perhaps a little fast over this, but these are other data, and you can ask me if you're particularly interested. But what we're looking here is that we're looking at gene expression, and what we can see here is that not all the genes in these are 3 genes in an organism in a bacterium, and they are being activated by growth on different substrates, where these ones will have a significant amount of linkages with lignin, between lignin and xylan. And you can see that not all the enzymes are being activated, which means that not all enzymes within this family have the same function also in biology. And here you also see that this is an experiment that is more to do with biotech. We are throwing to a mixture of corn cob biomass. Is basically mashed up corn cobs. We are throwing a cocktail of enzymes called ultraflow, and then we are seeing if addition of these enzymes actually helps. we can see that we're getting more glucose released when we are adding some of these C15 enzymes, but it's not equal. So there are differences. So if we look at the mechanism very briefly now, we can see that we can divide it up into an acylation step and a deacylation step. And we have this catalytic nucleophile that attacks the bond, the ester bond. And here we would have the sugar, and here is just a methyl group to represent the rest, where lignin would be. So we have the histidine abstracting a proton, so that the serine becomes a beta nucleophile. We form a tetrahedral intermediate with a charge here, which needs to be stabilized by what is called the oxyanion hole. And these acid groups, which we have two in this particular enzyme, they will stabilize the charge here, and that's how they're supposed to help. And then we form a covalent intermediate on the enzyme, and this is the acylation step. And in the deacylation, we go through a similar process where we then can promote the attack of the covalent intermediate by water, and then release the second part of the product. And we have probed the role of the acid residues, which are a bit interesting, because we see that, as I said before, for the structure that I showed you before, the fungal structure, that we have the acid at a different place than in the canonical enzymes, but some of the bacterial ones have it on the beta strand where all the other, more or less, alpha beta hydrolases have. So what we see here is that we have an enzyme that has both, and must be the intermediate in evolution between the normal alpha beta hydrolases and this unusual position of alpha beta hydrolases. And why do I say that? Is because if during evolution I actually just have removed the one catalytic acid, I end up with a presumably dysfunctional enzyme that is going to be removed by evolution. But if first I duplicate and I generate this intermediate enzyme, which has got both the acids, then I can end up with removing this and having still a functional enzyme. Right, I can see that we're going to run out of time, so I'm going to just skip through some of this and tell you that we've probed the role of both the serine and histidine, that of course are very important, but also these acids, these two acids in this particular enzyme, and we probed them by QMMM calculations, which I'm not going to go into much. I'm just going to show you this little video, but basically this allows us to actually study each step, the acylation and the deacylation individually. And what we can see then, to cut a long story short, is that both are important in the calculations, and they're particularly important for the deacylation step. And this we can also see if we do the experiment and we mutagenize, but of course then we do not know if we have an effect on the acylation or the deacylation step. But you can see that the kcat for the mutants where we have removed the acid are lower than the kcat for the native enzyme. Then we probe the oxyanion hole, where in this case, unusually for an alpha beta hydrolase, we think we have an arginine residue that is contributing to stabilizing this intermediate. And then what we've shown here is that from the calculation, we can see that the acylation step is almost, it has a huge barrier. And indeed, we see that we can do an experiment in the crystal and actually trap a covalent intermediate because the enzyme gets stuck here, and it then gets blocked at this stage. So I think I only have a couple of minutes if I allow myself to have a couple of minutes. So I want to just try and make another very important point about this, is that it's all very well we can access by things like computational methods, reactions with small substrates, but what we're interested here is reactions with biomass. And the first information, structural information, was obtained with this fungal GE with a very small product. So it's just basically is with the glucuronic acid. So we have some information about the close interactions in the active site. But what I think has been also very important, we can see the interactions here in a second. You see we have all these hydrogen bonds that are keeping the glucuronic acid firmly in the active site. But what has been very important to demonstrate is that we have interactions with some larger fragments of the plant cell wall, because it's not forgiven that these enzymes might have specificity for the actual plant cell walls. A lot of esterases are very unspecific, but I think I've shown you that these are not unspecific, and we can see here in our structure that we have a considerable interaction with the backbone xylan as well, and presumably, if we had the information, also with side chains on the xylan. And similar results were obtained also by our next door neighbor, Sine Larsen's group, in collaboration with DTU, with a fungal enzyme, but we've been working primarily on these bacterial ones. So we know that it interacts with the carbohydrate portion of the biomass. What about the lignin part of the biomass? Well, here we only have circumstantial evidence because we've never been able to actually trap in a structure an intact substrate, no matter how much we have tried. So we've done some docking, we've done some mutagenesis. When we say we, I mean the mutagenesis and those functional studies primarily in Johan's lab, but we can't obtain a Michaelis-Menten complex. And finally, in this, what might seem to you as a little bit perhaps as stamp collecting, we've also found that there's a lot of differences in the loop structures and the overall structures of these enzymes. So if this is the fungal one, we have we can see that in many of the bacterial members we have extra parts, extra loops that fill quite a lot of space. And this is another example where we also have an N-terminal extension. And this is the latest example. It's not quite obvious here. This is also bacterial. It doesn't have these extensions, but it has a very protruding region too, as we have named it, which is obvious if you turn it around. It's really poking up. And here are some aromatic residues that could interact with lignin, but we just don't have the evidence for that yet. And again, we have to think that these enzymes are going to have to attack something that looks like this. This is a model of a more realistic model of what they actually are sitting on, with cellulose and xylan and glucuronic acid. So this doesn't have the lignin, but it has the polysaccharide part that you has been working with. And obviously, any protrusions, any big loops that are here are going to make a huge difference to how these enzymes can interact with the complex biomass. And now I've taken really far too much of your time, but just as perspective, we are working really hard on looking on the structural basis of specificity of more complex substrate. We also want to try and improve the stability from what we've learned from the hyperthermophilic enzyme. We want to try and understand why some of the enzymes that should be active, we do not find any activity for. So basically make sense of things as scientists do. And I must also acknowledge some of the pictures I've nicked from the web. So I do that. And thank you again.